Good morning, everyone. Has been our day. We thank God for this great day. Thank God for what God has been doing and what God is doing in our midst. We thank God for the last webinar. It was a great success. We received a lot of response from a lot of people. And I'm sure that few among those people are also here this morning for the second edition of this webinar. It's a great news that is coming to us at this very period. And we appreciate God for what God is doing in our midst. I want us to sit back and enjoy the seminar and prepare our questions, because today we are not in a hurry to leave this place. Prepare all your questions and every question will be attended to. So as we await others to join us, we'll have to kickstart the webinar. I want to introduce our doctor, Dr. Ogochukwu Obona. Is going to be taking us today on genetic therapy for the permanent treatment for sickle cell anemia from UK. Want to welcome our doctor. Dr. Sir, you're welcome, sir.
God Thank bless you, Mr. Ayo.
The floor is open for you.
Thank you. Thank you, our noble guests. Thank you for families. Thank you for warriors. This is our second series in an outstanding planned out sessions for these webinars dealing exclusively on hemoglobinopathy. Hemoglobinopathy are those conditions that are based on the defaultive gene mutations at the hemoglobin level of the blood. Sickle cell anemia is one of them. Thalassemia of various sorts are some of them. In the last episode, our first series of the webinar, we explained this, but for the benefit of the ones that we are not there, we will just rehash on that in terms of the explanation of sickle cell anemia and thalassemia. Sickle cell anemia is when the blood, the hemoglobin in the red blood cell, produces some sickled blood cells, which are blood that are in crescent shape instead of the round spherical shape. Those crescent shaped blood cells are usually sticky. They don't last for long. Regular red blood cells that are round and spherical last up to 120 days in the body. These sickled ones can last anywhere between 8 to 12 to 18 days. And when they die, they block the pathways in the small blood vessels carrying blood around the body. That is sickle cell on its own. It comes up with a whole lot of other secondary manifestations. Again, thalassemia, on the other hand, is when the same hemoglobin is not present, or when it's present, it doesn't produce enough hemoglobin. That is what we call thalassemia. So you can understand that with respect to thalassemia, the body doesn't have enough of the hemoglobin to transport oxygen around the body. In some instances, it's absent. When there's shortage of that, you can understand that the body function will not be optimal, and the person is tired all the time for lack of oxygen. Hypoxia sets in, and other conditions. We're just scratching a little bit to rephrase and refrain on the subject matter thalassemia and sickle cell anemia. Today, our major focus is on the treatment of this hemoglobinopathy, as we call them together. The treatments vary. For as long as mankind has been going through this, there has been every form of palliative until modern science now went in deeper to try to find lasting solutions to that. Today, gene therapy is the leading pathway to the permanent treatment of this. What is gene therapy? Gene therapy is a treatment, formulation of medical treatment that is geared towards going to the genes and the chromosomes to be able to correct this defect. Sickle cell anemia, thalassemia, the entire gamut of hemoglobinopathy rests on the mutation at the genes. So to treat it means that you have to go to the gene to correct it. How do you go to the gene to correct it? Going to the gene is like going to space or going to another planet beyond space. Getting to that, I'm using space technology to illustrate what gene therapy is. Sometimes you can hear scientists call cell therapy or gene therapy. Simple genetics teaches us that the genes are embedded in the chromosomes. The chromosomes are in the cells. So any treatment that will go to the cell to treat a condition is called cell therapy. Cell therapy, if you now hone closer to the chromosome and to the gene, that is what you call gene therapy. Basically, with that understanding, we've been having other forms of treatment. Why is gene therapy different from other forms of treatment? Some other forms of treatment may just be at the blood level or at the organ level. Which organs are responsible for this in terms of treatment and cure or palliative? We know that the kidney makes blood as well. We know that the spleen stores blood as well as destroys red blood cells, especially the defective sickled blood cells. But bottom line, the kitchen of the body where blood is cooked, I'm using the word cooking because it's production, is at the femoral head, the long bone of the femur that goes to the bone marrow. So the bone marrow is essentially the hotspot where most of these activities take place. And where do we get the bone marrow? Bone marrow is in between the long bones of the body, especially the femur being the thigh bones. The hands are also part of it, the arms. But most often than not, we gear towards the bone marrow at the femur. To get to the bone marrow, you need vector agents that take the therapeutic agent into the bone marrow. What do you get at the bone marrow? You get essentially stem cells, meaning cells, the basic cells of life that now turns on through genetic processes or biological processes. Enzymes are involved. Some other hormonal activities are involved that get to the bone marrow and switch it on to produce either complete red blood cells or to switch it off from producing the sickled red blood cell, or to tune in on again to produce more of the hemoglobin that may be absent in thalassemia. So this gamut of activities in hematology, meaning dealing with blood, lies essentially at the bone marrow. In nature and at birth, every one of us do have so much of these stem cells. These stem cells are precursor, the basic cells that sustain life. Upon switching on, they can now turn into specialized cells like red blood cells, white blood cells, platelets, and the rest of them. Besides red blood cells, there are some other cells in the body, the nerve cells, which we call neurons. They also start from basic cells, which are also called the basic stem cells. Every body, every tissue, every cell in the body have their own names and their own functions. But basically, stem cells are just, it's like giving you a blank slate to write on. Nature presents us with that option of being able to understand how the body works and for the scientists to be able to manipulate these stem cells in getting the result that we target. And that is full red blood cells, devoid of the Sickle ones. We try to manipulate it through some other treatments in surgical cases, like we do the bone marrow transplant, remove the defective cells from the bone marrow and transplant it with very correct ones, and now trigger it, switch it on to now start producing normal red blood cells. That's a procedure. Today, science have gone way deeper. We now have some other medications. I'm being careful in not calling names of some of these medications that are also gene treatments. They come very expensive. Some of them come as much as $3.5 million just for one treatment. Some of them come as much as $2.8 million. If you Google most expensive drugs, most expensive gene therapies, or gene therapies available for sickle cell disorder, you will see the names and the prices of this. Why we are very much excited today is because not just the cost and the price of those gene therapies, justifiably, they spend so much in the research and development of those gene therapies. You can hear today the buzzword in the medical community is gene therapy, because once you get to the gene, everything will be corrected. Getting to the gene, I have talked about space technology. You take off from Earth with a rocket. It could be NASA, it could be Elon Musk SpaceX, it could be Richard Branson, it's Virgin Galactica. You need that vector to shoot off from where you are launching it. It carries the payload, meaning what you want to deliver to space or to the other planets. Once it gets above the atmosphere, the rocket, being the shuttle carrying it, drops. The payload with another rocket continues. It's being programmed to work with the pressure, which is gravity. It breaks gravity, gets into another level. Once it gets to a certain level, those rockets will also give way and the payload will continue the journey. That is what gene therapy can be likened to. With respect to gene therapy, the payload is the active ingredient that makes these things, that does the correction, that edits the defective gene or the mutated gene. The moment you take it in, it is built with these rockets, multiple layers of rockets that penetrate into the bloodstream the moment it gets into the mouth. it moves from the mouth or the gastrointestinal system so that it will not be digested there into the bloodstream. From the bloodstream, it gets to certain specific organ sites. That rocket drops off. Another rocket carries it in, which we call vector, carries it in to certain areas, like we mentioned the femur, the long bones, to the femoral head. That one now gives way, carries the payload, which is the active ingredient, straight to the gene site by way of what we call the telomere at the end of the chromosomes that houses the genes. So gene therapy is a very deep science. You have so much layers, and the cost of licensing each of those layers by scientists who discover these things is enormous. That is why when the big companies and smart scientists around the world collaborate, they know which firm has developed one, they know the other one that has developed the other vector, they know the ones that have developed the active ingredient, which is a curative agent itself. Now, you can now assemble them, aggregate them, and develop the most awaited drug, which is the gene therapy. We're a little bit careful in trying to use the regular words we all know. If I'm talking to a community of scientists, I will be very careful in saying drug. I may use the word medication. I may use the word therapy. I may use the word remedy. To the layman, all of these things, what they're asking for is treatment and cure. To the scientist, the moment you use drug, you are talking about that same remedy, that same therapy that is formulated with synthetic chemical. To another scientist, if I use the word biologics, he will understand that biologics means those ones that are more like potent, therapy-graded natural substances, like enzymes, like hormones, like extracts. These extracts, whatever you want to call them, it is all grouped as biologics. They come very sensitive and expensive. Expensive because you don't have preservatives in them. You make them, you tailor-make them based on request and demand. And their lifespan is very short. You request for, it's like the way they make vaccines. Vaccines are batch produced. You make them in batches. You don't make them the way you make chloroquine or any of these other synthetic drugs. You don't make them to keep over the shelf. You make them and store them in refrigerative platforms. The same way you harvest organs and carry organs immediately, an aircraft is waiting, you deliver it to the site of the hospital where transplantation will take place. That's basically the same way we handle these biologics, because they contain live enzymes, they contain live hormones, they contain live cells. You will agree with me that the process of logistics alone, besides the research and development, makes these things, each of the layers, come with added financial value on it. So when you hear about gene therapy today, you will now classify, could this be as a result of this, or is it from the foundation of this or the other components? But bottom line is that gene therapy are those therapies that go to the gene level or cellular level to correct any anomaly, most of them mutations. We have also seen that the invasive therapies, meaning surgical conditions, surgical processes that we use to correct some of these things. In our last webinar, we talked about in vivo and we also talked about in vitro. meaning in the test tube, or in vivo in the body. The subject of our discussion today is hemocytic rage or hemostim. That is the gene therapy that Genetics have the best in class of delivering for the treatment of this hemoglobinopathy. Why do we call it best in class? We call it best in class because it's the only one that is oral. It's the only one that is in vivo, meaning inside the body. It's the only one that upon taking it, it gets straight to the organ sites and it gets to the genes and it reverses the gene on one-time treatment of 15 days. It's not the ones that you go and harvest from a compatible sibling or somebody who have the same size. In that way, the gene therapy by Genetics stands head and shoulder above the other ones. We will also come to cost. Why we say is the first and the best in class is because the other ones, you are talking about millions of dollars. The other ones, we are still watching and seeing the long-term effect of it. Long-term in the sense of the efficacy. We have come across some of these procedures where you need to look for matching stem cells or tissue type to be able to transplant into the recipient. Sometimes some of them work up to 50%. Sometimes some of them work and after a while you have a relapse. From every case that this hemostim or hemocytic rage have been presented with, we have a continuous success rate of 100%. So the gene therapy is not the only way that treatments can be carried out, but it is the most sustainable way for treatment. Everybody who is associated with carriers, with warriors, will attest that they have tried different treatments. They have tried herbal treatments. They have tried synthetic chemical treatments. They have tried ameliorative treatments. They have tried physiotherapy. All of these things are geared towards helping the warriors. All of them come with the best intention. But what we are talking about here is the success rate. It's not just about hemoglobinopathy, sickle cell, and thalassemia. The application of gene therapy also goes to cancer cases that have gene origin. It goes to other conditions that we associated with defective genes. However, our core area in this hemoglobinopathy, meaning diseases of the hemoglobin, is that it restores. We do have a technology, which is one of the treatments that is considered as CRISPR, meaning gene editing, where you have to harvest the same stem cells or harvest the blood of the recipients, take it out, correct it in the test tube, and transplant it back into the bone marrow. It does work, but it is very selective. It is very tedious. It is ongoing. You cannot use that to massively lay out that same technology and skill level to address the millions, the tens of millions of warriors all over the world. Thalassemia alone, for people living in Africa, predominantly, they go through the sickle cell anemia of one sort or the other. It could be the homozygous one, where their blood is HBSS, hemoglobin SS, being the sickle cell. It could be the HBSC, another variation of single gene from the father or mother, being the S1 and the C1. We call that one heterozygous sickle cell. It could be some other variants of the thalassemia. But in Asia, particularly as Nigeria is the world capital for sickle cell anemia, so is India the global capital for thalassemia. Some of the Africans also do present with thalassemia, but not as much as Indians. If you look into the whole origin of the thing, it is still basically the same thing. Mutation in terms of evading a long time ago from the menace of malaria parasites. That was the origin of this same mutation. Trying to evade malaria. Trying to evade malaria, mankind ended up mutating that gene. It was a defense mechanism, but it has come back now to affect the offsprings and descendants of the same Afro-Caribbean ancestry and a few people who may have had in the past their origins or their ancestry in Middle East, in Mediterranean area, in Latin America, in North America, US and Canada. All over the world, we have semblance of this manifestation. So essentially, the modality for delivering of this gene therapy of hemostim, we call it hemostim HS 1.0, is that I have discussed about the cold chain delivery system in terms of biologics, which it is. We are now talking about the need for preservation. We have, in the past webinar, presented a copy of this so that for those that have not seen it and have no idea of what it is like, I will show you what a sample of it is. 1 second, please. This is the hemostim yet again. It is frozen. In that way, it will preserve its integrity. Every part of the components of this is direct extract from phyto background, phyto origin. Some of them are stem cell, phytostem cell from plant origin. When we talk about stem cell, please, let's not forget that there are two classes of stem cells because there are two classes of life. Since stem cells are the basic cells in life from inception, we have animal lives and we also have plant lives. In the classroom, we have plant genetics and we also have animal genetics. Most people who hear about stem cells, they think that it comes from embryos, which we call embryonic stem cells. So some people do ask us, how can one drink embryonic stem cell when it comes from embryo? The embryo coming from placenta of newborn babies or newborn lives. Is that edible? We said, no, that is not where we are coming from. We have advanced to where we now can harvest the plant stem cell because plant has DNA of its own. The DNA of plant is different from the DNA of animals, which human beings, we are part of the animal kingdom. But plants are there right from origin of mankind to sustain animals. We may as well use that with better insights, the same plant stem cells, to formulate and direct it to serve us. We are not in argument of plant kingdom supporting animal kingdom in terms of nutrition, in terms of therapeutics. And here we are today. Science have gone deeper. We have researched to find out. Earlier on, we believe that embryonic stem cells are stable. You can formulate things with embryonic stem cells. We believe that plant stem cells or phytostem cells were unstable and cannot last long. Talk of using it for therapeutic graded. However, today, we have successfully succeeded in using plant stem cells, which is the phytostem cells, stabilize it, formulate drugs with it. Sometimes we also build up direct extracts of certain hormones. Plants do have their own hormones, not just humans. Our innovation in this class of therapeutics happens to be why we call our product the best and the first in class. All other forms of gene therapies are very much either based on synthetic chemical formulations or combination of that and the embryonic stem cells. We are not part of that. We are 100% of phyto stem cells. Therefore, that warrants us the access into the body for GME. Okay, so it warrants a higher degree of safety, efficacy, and successful 100% therapeutic results. We will now develop our discussion based on questions and feedback. So, Mr. Ayo, please choose the audience to ask questions because we may have elaborated enough on the gene therapies and the available treatments. Any other questions, we will now take it from